simply these little kidney bean-shaped organelles that we all think of. In fact, in Vamsi's early slides, he showed us a great video of mitochondrial fusion and fission. So mitochondria are really these dynamic organelles. They fuse, they fission, they communicate with the rest of the cell. And I think much of the work that has revealed these phenomenon has come from Jyoti's group. So Jyoti has some Midwestern roots. She, having done her degree in chemistry at the College of Worcester in Ohio, she did her doctoral work in pharmacology at Vanderbilt, and then was a postdoctoral fellow with Peter Walter out in UCSF. So I'm mentioning that we're realizing that mitochondria have all these other connections to the cell. In fact, for years, if you tried to purify mitochondria, you would be very annoyed by the fact that you couldn't get rid of the endoplasmic reticulum. And it turns out that it's not just a contaminant. There's actually really, they're tethered in some meaningful way. And in fact, through some really elegant work, Jyoti's group has shown that the ER actually participates actively in mitochondrial fission. She's also characterized something called the Mekos complex, which I think she'll probably tell you about, which is very important for forming those cristae structures that we're all very familiar with when we think of mitochondrial morphology. So Jyoti, among many other awards, has been recognized by a Keith R. Porter Fellowship and by UC Davis Faculty Research Lecture Award. She's also very active in many other areas of science. She's a member of the Scientific Advisory Board for the Max Planck Institute for the Biology of Aging, the American Society for Cell Biology Public Policy Committee. She's also on the editorial boards of eLife and Traffic, and is a senior editor for the Journal of Cell Biology. Apparently also likes to ride horses quite a bit. So, Jyoti?
Where's the pointer? Yeah, I did. I grew up in Cleveland. Great, thank you. Thanks for the nice introduction, and I'm also honored to be here with my distinguished colleagues. And here at Madison, this is my first time here, except when I was a child, when we used to vacation in the Dells. That was our big treat from Cleveland. I have very fond memories of that. So I love Wisconsin. I love cheese. Come on, that had to happen, right? So, as David told you, my lab is really interested in the relationship at its very fundamental level between mitochondrial form and function. So we're really interested in how mitochondria look, why they look that way, why the way they look is important for their function. And I guess the way I've characterized that over my career is in this question here. How is mitochondrial behavior controlled? And by behavior, that means to me, and it means to my lab, a lot of different molecular determinants that essentially cooperate to give rise to this beautiful organelle, the structure that you see here. And this particular cell type is the budding yeast, Saccharomyces cerevisiae, and it's one of the model systems that we use in my lab to study the relationship between mitochondrial form and function. I like this system not only for the power of the genetics that it has, but in the sense that it's a real in vivo system. And it's allowing us to understand how the individual determinants that give rise to this snapshot, if you'd like, of the structure that you see, kind of communicate with each other on a systems level to produce this picture. And so it's given us an opportunity to really know mitochondrial behavior in a very deep way, which is what I'd like, at least in part, to share with you. And so these determinants that I'm referring to are ones that we've characterized in my lab and other people have in theirs over the years, and they include mitochondrial dynamics, which is, I guess, what I'm somewhat famous for. That includes mitochondrial fusion and mitochondrial division, the actual machines that do that. There are active mechanisms to position mitochondria, and again, in this particular cell type, the position of this organelle is right at the cell cortex. So it's right there underneath the cortical ER and plasma membrane. There's, of course, machinery that moves it around. And again, in this organism, it's all actin-based, but in human cells, it's a combination of actin and microtubule-based motilities. How it looks in terms of its internal structure. So everybody appreciates that mitochondria have two membranes. The inner membrane, it has a very distinct shape and is laterally differentiated. And unfortunately, I'm not really going to have time to talk about what we know now about the machinery that does that. But it is an important determinant of how it looks on the outside too. Just know that. And then finally, this last but not least one is really what got me interested in mitochondria to begin with. The fact that it has its own chromosome. It's an organelle that has its own chromosome. How cool is that? Must be important. And you can see that this chromosome is organized into its own organelle in the innermost compartment. We call it nucleoid out of respect for its origins. And in fact, I hope you appreciate that within this reticular-like structure, not bean-shaped, as David told you, these are nicely spaced out, not only throughout the cell, but within this reticulum. And so there are mechanisms in place that determine the spacing, and also the ability to faithfully inherit the mitochondrial chromosome from mother to daughter. And this mechanism is largely still unknown, unbelievably. And it's what still drives my lab. So we're always paying attention to it when we look at all the rest of the molecular determinants. And that'll hopefully come out as I go along to tell you about some of the things we've learned over the years. Now, about that chromosome, I consider mitochondria pivotal to the evolution of eukaryotes. Right about the time, we all come from the same thing. Everybody knows this, right? There's 3 kingdoms of life on this planet. And where we cut out over archaea is exactly the point in time where we acquired this endosymbiont that became mitochondria. And as Vamsi said, even though we have the chromosome, it's not very big anymore. A lot of the genes have either been lost completely or lost to the nucleus. But what that has enabled us to do, it's been transformative in evolution of eukaryotes. Because this little chromosome is small, so it's easily managed. But it's in multiple copies, and it's still linked to the membrane that creates the energy for the eukaryotic cell. And it's by that design that enabled us to amplify our genome and have rarefied abilities to walk and talk and do all those good things. So I consider mitochondria to be the pivotal point in the evolution of eukaryotes. Now, as Vamsi said, even though mitochondria has evolved from a bacterial ancestor, and I stole this from one of his reviews because I knew I was going to follow him, a lot has happened since then. In fact, as I said, many of the genes in the original endosymbiont are believed to be completely lost. Some of them, most of them,